Why do almost 4 out of every 10 Indians share the exact same rare blood type that barely exists in Europe? And why does that number get bigger the closer your ancestors lived to Punjab, and smaller the closer they lived to Kerala? If you enjoy this kind of deep dive into where we really come from, hit, like right now, subscribe if you're new, and send this to one person in your family who has B positive blood, because by the end of this video, they will never look at their blood test the same way again. Most people think a blood type is just a medical detail, something a nurse writes on a form, something you need to know before surgery or before donating blood. That is all true, but it is also the smallest part of the story. Your blood type is written into your DNA. It did not appear yesterday. It did not appear with you. It was carried to you across thousands of generations, through famines, through migrations, through diseases that wiped out entire villages, through empires rising and falling. Every person who carried your exact blood type before you had to survive long enough to have children. And so did their parents, and their parents, all the way back. So when scientists tell you that B positive blood is unusually common in India and unusually rare almost everywhere else in the world, they are not describing a coincidence. They are describing a signature left behind by a very specific and very ancient chain of events. And once you understand what that signature means, a random letter on your medical card turns into a map that leads back through the Mongol armies of the 13th century, through the crowded cities of the Indus Valley, through the horse-riding herders of the Central Asian grasslands, and all the way back to a single genetic accident that happened roughly 10,000 years ago. Let's start with the number itself, because the number is genuinely strange. In India, roughly 38 to 40% of the population carries B positive blood. That is not a small regional pocket. That is close to half the country. Now look outside India. In the United Kingdom, B positive sits at around 8%. In the United States, around 11%. Across most of Western Europe, it hovers around 10 to 12%. Think about that gap for a second. In India, B positive is almost the norm. In Western Europe, it is one of the rarer types you can have. Same species, same planet, same basic biology. So why such a massive difference? To answer that, we need to go inside the blood itself, down to the molecules sitting on the surface of your red blood cells. Your blood type is decided by a single gene, sitting on chromosome 9, called the ABO gene. This gene is basically an instruction manual for building a tiny chemical marker, an antigen, that sits on the outside of your red blood cells like a flag. If your body builds the A version of that flag, you are blood type A. If it builds the B version, you are blood type B. If it builds both flags at once, you are AB. And if the instruction manual is broken in a very specific way, so that no flag gets built at all, you are blood type O. The positive or negative part of your blood type, the Rh factor, comes from a completely different gene. It decides whether another protein called the D antigen is present on your cells or not. Most Indians, somewhere around 93 to 95%, are Rh positive. Rh negative is genuinely rare in India, far rarer than in Europe, which is exactly why Rh negative blood donors are treated like gold in Indian hospitals. But it is the B allele, the instruction for the B flag, that carries the real story. Genetically speaking, O is the oldest version of this gene. It is found at extremely high levels in populations that stayed relatively isolated from later migrations for tens of thousands of years, places like parts of the Americas, where O blood type can reach 75% or higher in some indigenous groups. O came first, long before anyone had a name for blood types. A came later as a mutation of that original gene. And B is the youngest of the 3 major versions. It appeared as a mutation of the A version, caused by a change in just 4 amino acids in the enzyme the gene produces. 4 tiny changes. That is the entire molecular distance between type A blood and type B blood. Everything else about you, your height, your face, your language, your family, sits on top of a difference that small. Scientists have actually managed to trace this mutation through ancient human remains, and this is where the story stops being theory and starts being evidence you can hold in your hand. A well-known study published in the journal, Scientific Reports examined blood type genes recovered from human skeletons found across Europe and Asia, some of them more than 40,000 years old, along with genetic material pulled from the bones of Neanderthals. What they found was striking. In the oldest samples, blood type O dominates almost completely. The B allele is either missing entirely or vanishingly rare. Then, sometime after the last ice age ended, roughly 10,000 years ago, the B allele starts showing up in the record. And it does not appear randomly across the map. It appears first and most strongly in one specific region, the high plateaus near the Himalayas, and the vast grasslands stretching across Central Asia. This is the birthplace of blood type B, not India, not Europe, the steppe. And once it appeared there, it did something unusual. It spread, fast and in one direction more than any other. If you draw a line on a map showing where B blood type is most common today, that line runs directly through Central Asia, through the historic steppe corridor, and down into South Asia. It gets weaker the further west you travel into Europe, and weaker again the further you travel into the deep south of the Indian subcontinent. That is not a coincidence. That line is the fingerprint of a migration, drawn in blood chemistry instead of ink. Around 4,000 years ago, between roughly 2000 and 1500 BCE, groups of pastoralist herders from the Eurasian steppe began moving south into South Asia. These were people who lived on horseback, who moved constantly with their cattle, and who eventually reshaped the language, the religion, and the genetic makeup of huge parts of the subcontinent. If you have ever come across deep ancestry videos discussing the Y chromosome marker R1A in the Indo-Aryan migration, this is the exact same population we are talking about. And they did not just bring their language and their cattle south with them. They brought their blood type. This is the exact same population we are talking about. And they did not just bring their language and their cattle south with them. They brought their blood type, alleles, too. Elevated B frequency was part of their genetic package, riding along quietly inside every single person who made that long journey south. Here is where it gets genuinely interesting. The parts of India that historically received the strongest wave of that steppe migration, the northern and northwestern regions, are today the exact same parts of India where B positive blood is at its highest, sometimes touching 45%. As you move south, away from the heaviest steppe influence, B frequency drops and O frequency climbs back up. Two completely different types of evidence. Blood samples collected in hospitals over decades of ordinary clinical work and cutting-edge ancient DNA sequencing done in modern research labs are pointing at the exact same historical event. When two unrelated methods, one old and mundane, one new and highly technical, agree that closely, you're not looking at a coincidence anymore. You're looking at something real, something that actually happened to real people a very long time ago. But the steppe migration is only one layer of this story. There's an older layer sitting underneath it, and honestly, it might be even more important than the migration itself. Before those steppe herders ever set foot in South Asia, the subcontinent was already home to sophisticated farming societies, including the great planned cities of the Indus Valley civilization. And genetic studies suggest that meaningful levels of the B allele were already present in South Asia before the steppe migration ever happened. So the steppe migration did not create the B allele in India out of nothing. It added to something that was already there. If you're finding this useful, do me a quick favor, tap that subscribe button and drop a comment telling me which Indian state your family comes from, because I genuinely want to see how closely the blood type map in this video matches the story of your own family. So if steppe herders were not the only reason B blood type became so common in South Asia, what else was going on underneath the surface? The answer takes us into one of the strangest and most fascinating corners of human biology, natural selection, acting directly on your blood, generation after generation, without anyone ever noticing it happening. Here is the idea, and it has a name, balancing selection. Sometimes a particular gene variant does not just survive by pure accident. It survives because it gives the people who carry it a genuine edge against disease. If that edge is strong enough and consistent enough over thousands of years, it can push that gene variant to unusually high frequency in an entire population, even if it started out as a rare, obscure mutation carried by only a handful of people. And blood type turns out to be deeply tied to how your immune system handles certain infections in ways that most people never learn about in school. Take cholera. People never learn about in school. Take cholera. The bacteria responsible for cholera produces a toxin that latches onto a molecule found in especially high amounts on the cells of people with blood type O. This is not a small statistical footnote buried in a research paper somewhere. Research from cholera outbreaks in Bangladesh and other parts of South Asia has repeatedly found that blood type O individuals are significantly more likely to develop the most severe, life-threatening form of the disease. In a river delta civilization, where cholera and other waterborne diseases were a constant and recurring threat, that is a massive disadvantage to be carrying in your blood. Blood type B, on the other hand, shows a different disease profile. Some studies point to reduced susceptibility to certain strains of E. coli, along with distinct interactions with the malaria parasite compared to type O. None of this means B blood type made anyone immune to disease, and no serious researcher claims that it did. It simply means that generation after generation in the wet, specific disease environment of ancient South Asia, hot, humid, densely populated, dependent on rivers for both drinking water and farming, people who happened to carry the B allele were, on average, slightly more likely to survive long enough to have children of their own. Slightly more likely does not sound dramatic on its own, but multiply a small survival edge across hundreds of generations, and the math becomes enormous. That small edge, compounded quietly over thousands of years, is genuinely enough, according to population geneticists, to explain how B blood type climbed from a rare mutation carried by a small group of steppe or Himalayan foothill dwellers to nearly 40% of an entire subcontinent. So by the time the steppe herders arrived around 1500 BCE, they were not introducing B blood type to a population that had none of it at all. They were adding a second wave of the B allele on top of a population that may have already been unusually rich in it, thanks to thousands of years of disease pressure in the crowded, river-fed cities of ancient India. Two forces, one ancient and biological, one later and migratory, pushing in the exact same direction at different points in history without either one knowing the other existed. And there's a third wave, layered on top of both of those, one that most people have never even heard connected to their own blood type. In the 13th century, the largest land empire in human history exploded out of Central Asia, the Mongols. Under Genghis Khan and his successors, Mongol armies swept across almost the entire Asian continent, reaching from the Pacific coast all the way to the edges of Central Europe, conquering kingdoms and empires that had stood for centuries. Those armies did not just carry weapons and horses with them. They carried the genetic legacy of the Central Asian steppe, the very same region where the B allele had first appeared thousands of years earlier, now concentrated even further by centuries of intermixing among steppe populations across a vast stretch of Asia. Mongol forces pushed into the Indian subcontinent multiple times between 1221 and 1306 CE. They never conquered Delhi outright, and the Delhi Sultanate held its further by centuries of intermixing among steppe populations across a vast stretch of Asia. Mongol forces pushed into the Indian subcontinent multiple times between 1221 and 1306 CE. They never conquered Delhi outright, and the Delhi Sultanate held its ground against them in several major battles. But the Mongols still left a real and detectable mark, particularly in parts of northern India and Bengal, where their raids and incursions were most frequent. Along with that contact came another small injection of steppe-associated ancestry, layered directly on top of the older Indo-Aryan steppe migration from nearly 2,000 years earlier. So when you look at a modern Indian medical record that simply says B positive, you are actually looking at the compressed final result of three separate historical forces: an ancient disease-driven selection pressure inside the crowded world of the Indus Valley, a massive Bronze Age migration of pastoralist herders from the Eurasian steppe, and a medieval wave of contact tied to the Mongol expansion, 7 centuries later. Three completely different eras, three completely different sets of people, most of whom never knew of each other's existence, all of them, across thousands of years, quietly pushing the same tiny genetic switch in the exact same direction. Now, let's zoom into the map of India itself, because the regional pattern here is almost too perfect to be a coincidence. In Punjab, Haryana, Rajasthan, Uttar Pradesh, and Gujarat, states that historically absorbed the heaviest dose of steppe ancestry, B-positive frequency regularly sits between 38 and 45%. These are also the regions where the Y chromosome marker most closely tied to the steppe migration, R1A, appears at its highest levels in modern populations, and where the cultural memory of the Vedic tradition runs deepest and oldest, move south through Maharashtra and into Karnataka, Tamil Nadu and Kerala, and the numbers begin to shift in a very consistent way. B frequency drops toward 30 to 35%, while O frequency climbs through Maharashtra and into Karnataka, Tamil Nadu, and Kerala, and the numbers begin to shift in a very consistent way. B frequency drops toward 30 to 35%, while O frequency climbs steadily upward. This mirrors, almost exactly, the well-documented decline in steppe-related ancestry, and the corresponding rise of the much older indigenous South Asian ancestry component, as you travel from the north down toward the southern tip of the subcontinent. Then there is the northeast of India, Assam, Manipur, Mizoram, Nagaland. These states also show elevated B blood type frequencies, but for a completely different historical reason than the Northwest. Their populations carry substantial Tibeto-Burman ancestry, tied to ancient migrations from East and Southeast Asia. A part of the world where B blood type is independently common shows a different disease profile. Some studies point to reduced susceptibility to certain strains of E. coli, along with distinct interactions with the malaria parasite compared to type O. None of this means B blood type made anyone immune to disease, and no serious researcher claims that it did. It simply means that generation after generation, in the wet, specific disease environment of ancient South Asia, hot, humid, densely populated, dependent on rivers for both drinking water and farming, people who happened to carry the B allele were, on average, slightly more likely to survive long enough to have children of their own. Slightly more likely does not sound dramatic on its own, but multiply a small survival edge across hundreds of generations, and the math becomes enormous. That small edge, compounded quietly over thousands of years, is genuinely enough, according to population geneticists, to explain how B blood type climbed from a rare mutation carried by a small group of steppe or Himalayan foothill dwellers to nearly 40% of an entire subcontinent. So by the time the steppe herders arrived around 1500 BCE, they were not introducing B blood type to a population that had none of it at all. They were adding a second wave of the B allele on top of a population that may have already been unusually rich in it, thanks to thousands of years of disease pressure in the crowded, river-fed cities of ancient India. Two forces, one ancient and biological, one later and migratory, pushing in the exact same direction at different points in history without either one knowing the other existed. And there is a third wave, layered on top of both of those, one that most people have never even heard connected to their own blood type. In the 13th century, the largest land empire in human history exploded out of Central Asia, the Mongols. Under Genghis Khan and his successors, Mongol armies swept across almost the entire Asian continent, reaching from the Pacific coast all the way to the edges of Central Europe, conquering kingdoms and empires that had stood for centuries. Those armies did not just carry weapons and horses with them. They carried the genetic legacy of the Central Asian steppe, the very same region where the B allele had first appeared thousands of years earlier, now concentrated even further by centuries of intermixing among steppe populations across a vast stretch of Asia. Mongol forces pushed into the Indian subcontinent multiple times between 1221 and 1306 CE. They never conquered Delhi outright, and the Delhi Sultanate held its further by centuries of intermixing among steppe populations across a vast stretch of Asia. Mongol forces pushed into the Indian subcontinent multiple times between 1221 and 1306 CE. They never conquered Delhi outright, and the Delhi Sultanate held its ground against them in several major battles. But the Mongols still left a real and detectable mark, particularly in parts of northern India and Bengal, where their raids and incursions were most frequent. Along with that contact came another small injection of steppe-associated ancestry, layered directly on top of the older Indo-Aryan steppe migration from nearly 2,000 years earlier. So when you look at a modern Indian medical record that simply says B positive, you are actually looking at the compressed final result of three separate historical forces: an ancient disease-driven selection pressure inside the crowded world of the Indus Valley, a massive Bronze Age migration of pastoralist herders from the Eurasian steppe, and a medieval wave of contact tied to the Mongol expansion, 7 centuries later. Three completely different eras, three completely different sets of people, most of whom never knew of each other's existence, all of them, across thousands of years, quietly pushing the same tiny genetic switch in the exact same direction. Now, let's zoom into the map of India itself, because the regional pattern here is almost too perfect to be a coincidence. In Punjab, Haryana, Rajasthan, Uttar Pradesh, and Gujarat, states that historically absorbed the heaviest dose of steppe ancestry, B-positive frequency regularly sits between 38 and 45%. These are also the regions where the Y chromosome marker most closely tied to the steppe migration, R1A, appears at its highest levels in modern populations, and where the cultural memory of the Vedic tradition runs deepest and oldest, move south through Maharashtra and into Karnataka, Tamil Nadu and Kerala, and the numbers begin to shift in a very consistent way. B frequency drops toward 30 to 35%, while O frequency climbs through Maharashtra and into Karnataka, Tamil Nadu, and Kerala, and the numbers begin to shift in a very consistent way. B frequency drops toward 30 to 35%, while O frequency climbs steadily upward. This mirrors, almost exactly, the well-documented decline in steppe-related ancestry, and the corresponding rise of the much older indigenous South Asian ancestry component, as you travel from the north down toward the southern tip of the subcontinent. Then there is the northeast of India, Assam, Manipur, Mizoram, Nagaland. These states also show elevated B blood type frequencies, but for a completely different historical reason than the Northwest. Their populations carry substantial Tibeto-Burman ancestry, tied to ancient migrations from East and Southeast Asia, a part of the world where B blood type is independently common, entirely separate from anything happening on the Central Asian steppe. So Manipur and Punjab can end up sharing a similarly high B frequency today, while their ancestors took two completely different journeys. one arriving from the northwest across the steppe corridor, the other arriving from the east across the Himalayan foothills and river valleys, to land on the exact same genetic destination, centuries apart. There's a similar story hiding in the western state of Gujarat and in Maharashtra, regions that sit right at the crossroads of these ancient movements. Both states show blood type patterns that blend features of the northern steppe, influenced pattern with features of the older, more southern pattern, which fits neatly with their geographic position as a kind of genetic border zone between the north and the south. Ancestry, it turns out, rarely moves in a straight, clean line. It moves the way water moves across a landscape, pooling more heavily in some places, thinning out in others, blending at the edges. Your blood type alone cannot tell you which of these journeys your own ancestors took. That level of detail really needs a full DNA test, one that looks at your Y chromosome line, your maternal line, and your overall ancestry percentages together. But your blood type is often the very first hint, a visible, testable clue sitting quietly on a piece of paper in a hospital file, pointing back toward one of these ancient roads, decades or even generations before anyone in your family ever takes a genetic ancestry test. It is also worth being honest about something here. Population geneticists caution that broad ethnic or caste level averages, while genuinely observed in surveys and blood bank data, come from studies of varying size and quality, and individual results can differ quite a lot from any group average. A blood type map like this describes patterns across large populations over long stretches of time. It does not describe you personally, and it should never be used to rank or judge any single community, family, or individual. The real value of this story is in what it reveals about deep, shared human history, not in sorting people into categories. There is one more piece of this puzzle that deserves attention because it is genuinely one of the most counterintuitive findings in the entire field. It involves the Rh factor, the part of your blood type that comes after the letter, the part almost nobody thinks twice about. Ancient DNA research has revealed something surprising here. Both the early hunter-gatherers of Europe and Asia and the Bronze Age steppe herders who later migrated into South Asia carried the Rh negative allele at noticeably higher rates than almost any population alive on Earth today. In other words, the very same ancient steppe populations who helped push B blood type frequency upward in India were, at the same time, considerably more Rh negative than their modern descendants living in the same regions today. So why is Rh negative blood now so rare in South Asia, sitting at only around 5 to 7% compared to roughly 15% in populations of European descent? The likely answer involves a medical condition called hemolytic disease of the newborn. When an Rh negative mother carries an Rh positive baby, her immune system can, in certain pregnancies, produce antibodies against the baby's blood cells, sometimes with serious consequences for that baby or for pregnancies that follow it. In a population where Rh positive blood is already overwhelmingly common, this creates a slow, steady, generation by generation disadvantage for the Rh negative allele. Over roughly 4,000 years since the steppe migration first arrived, that quiet disadvantage was enough to push Rh negative down to its current low level in India, while Rh positive became the overwhelming default across the entire subcontinent, including in the vast majority of Indians who also happen to carry the B allele. Before we go further, if a moment in this video actually surprised you, hit that like button right now. It genuinely helps this video reach more people who are curious about where their own ancestors came from, and it costs you nothing but half a second of your time. Now, here is something that makes this story even stranger. For most of human history, nobody even knew blood types existed. People had been transfusing blood between patients since the 1600s, and it kept failing in mysterious, sometimes fatal ways, with no explanation anyone could offer. It was not until the year 1901 that an Austrian scientist named Karl Landsteiner finally figured out why. He discovered that human blood came in different, incompatible types, and that mixing the wrong ones caused the blood cells to clump together dangerously inside the body. For that discovery, he later won the Nobel Prize, and modern blood transfusion medicine was born almost overnight. But Landsteiner had no idea, and could not possibly have known, that the very same discovery would one day be used by geneticists to trace human migration across tens of thousands of years. He was trying to solve a medical emergency happening in hospitals in his own lifetime. He had no way of knowing he had actually opened a window into the deep ancestral past of entire continents. It took another 100 years, and the invention of DNA sequencing technology, for scientists to realize that the same molecule responsible for saving lives on an operating table could also be read like a history book. Today, that history book is being read with tools Landsteiner could never have imagined. Researchers can now extract tiny fragments of DNA from a single tooth or a small piece of bone buried in the ground for tens of thousands of years and reconstruct entire genomes from it. They can compare that ancient genome against thousands of modern people from every part of the world and calculate, with real statistical confidence, how closely related an ancient skeleton is to a farmer in Punjab, a fisherman in Kerala, or a herder in Mongolia today. This is the exact technology that allowed scientists to trace the B allele back through the ages, from a mutation in an ancient enzyme through the steppe and into the modern Indian bloodstream. It is genuinely one of the most remarkable turns in the history of science. A man trying to stop deaths on a hospital table in 1901 accidentally handed future generations a tool to read the migration of entire civilizations, written quietly into the blood of everyone alive today. Think about the strange loop hiding inside that fact. Blood transfusions exist because of Landsteiner's discovery. Millions of lives have been saved because doctors now know to match donor blood to patient blood before a transfusion. And every single time that matching happens, in every hospital, in every city, in every country on Earth, it is quietly relying on the same molecular difference that also happens to be a 10,000-year-old fossil of human migration. The thing keeping patients alive on operating tables today is the exact same thing, letting historians reconstruct the ancient movement of entire peoples across continents. Few discoveries in the history of medicine have turned out to carry that much hidden weight. Let's step back for a moment and think about what kind of evidence we are actually dealing with here, because it matters. This is not folklore passed down through generations, and it is not a guess based on modern appearances or surnames. This comes from 3 independent lines of scientific evidence lining up with each other. Direct DNA extracted from human bones that are tens of thousands of years old, told to scientists by ancient teeth and skeletal fragments recovered from archaeological digs across Europe and Asia. Modern population genetics, built from studying the DNA of living people across hundreds of communities and comparing their Y chromosome markers, their maternal lineages, and their overall ancestry proportions. And decades of ordinary clinical blood typing records, collected quietly in hospitals and blood banks with absolutely no interest in ancient history at all, simply trying to match donors with patients. None of those 3 sources were built to prove a migration story. They were each built for a completely different practical purpose. And yet, when researchers lined them all up side by side, they told the exact same geographic story with a north to south gradient across India that lines up almost perfectly between blood banks and ancient bones. That kind of agreement, arrived at completely independently, is exactly the kind of evidence that makes scientists sit up and pay attention. So what does all of this actually mean if you are sitting there right now, holding a medical card that says B positive? It means your blood carries a compressed molecular timeline of South Asian history, folded into 2 simple words. Somewhere in your deep ancestry, there were people living in the river-based farming societies of the Indus Valley world, generation after generation, quietly surviving waterborne diseases that killed others living right beside them. Somewhere in your ancestry, there were herders moving south from the grasslands of Central Asia around 4,000 years ago, bringing cattle, language, and their own version of that same blood type gene with them on that long, dusty journey. And somewhere in your ancestry, there may even be a faint echo of contact with Central Asian populations during the age of Mongol expansion 7 centuries ago, carried forward quietly ever since. None of these ancestors knew they were carrying anything special. They were not thinking about blood types or antigens or amino acids. They were simply trying to survive floods, epidemics, wars, and famine long enough to raise children who could do the same thing all over again. But every single one of them, simply by surviving, passed forward that tiny 4 amino acid mutation that first appeared in the enzyme responsible for building red blood cell antigens, probably somewhere in the highlands near the Himalayas or out on the Central Asian plains, roughly 10,000 years ago, long before anyone had invented writing or cities or the wheel. It is worth remembering, honestly, that a blood type is not destiny, and it is not a complete biography. It cannot tell you your personality, your intelligence, your religion, or your family's exact village of origin. Scientists are still actively researching exactly how strong some of these disease resistance effects really are, and the full picture of ancient migration into South Asia is still being refined every single year as new ancient genomes get sequenced and new statistical methods get developed. This is genuinely active, ongoing science, not a finished textbook chapter sitting untouched on a shelf somewhere, but the broad pattern, the geographic gradient running from northwest to southeast, the connection to the steppe. Migration, the disease selection story running underneath it, is backed by a large and steadily growing body of published research, and it tells us something true about the deep past that almost nobody ever learns about in school. Every time someone in India writes B positive on a form, at a hospital, at a blood donation camp, on a college admission document, they are, without ever realizing it, writing down a fragment of a 10,000-year-old story, a story about a mutation that first appeared somewhere on the Central Asian steppe, a story about farmers in the Indus Valley who quietly outsurvived a deadly waterborne disease, generation after generation, a story about horse-riding herders crossing mountains and rivers into a new land they had never seen before, a story about an empire that once stretched from the Pacific Ocean all the way to the edge of Europe. All of it, every single piece of it, compressed down into 2 ordinary-looking words on a lab report. That is what makes this different from most trivia you hear about your own body. This is not a random fact you forget an hour later. It is a message carried forward across 10,000 years inside your own blood by every single ancestor who survived long enough to pass it on to you personally. Think about how many things had to go exactly right for that message to reach you. A single mutation had to happen in one person's DNA thousands of years ago instead of never happening at all. That person had to survive childhood in a world without hospitals or antibiotics. Their descendants had to survive famines, floods, wars, and outbreaks of disease that killed enormous numbers of the people around them. Somewhere along the way, some of those descendants had to leave their home on the steppe and walk, for months, sometimes years, toward a land they had never seen. And every single link in that chain, every one of them, without exception, had to live, long enough to have children. If even one link in that chain had broken, the mutation would have vanished, and you would not be carrying it today. You're not just a person with a blood type. You are living proof that an unbroken chain of survival, stretching back 10,000 years across steppe grasslands, river valleys, mountain passes, and empires, actually made it all the way to the present day. That is not a small thing to hold inside your own veins. So the next time you see B positive written on your own medical record or on your parents or your grandparents, take a second look at it. Really look at it. It is not just a blood type. It is one of the oldest and most honest records of where your family truly comes from, written not in ink and not in any language at all, but in the molecules quietly moving through your own blood right now as you watch this. If this video changed how you look at your own blood type, do 3 things before you go. Like it so more people get the chance to see this story. Subscribe so you don't miss the next one, because we are going even deeper into Indian genetic history very soon. And share it with the one person in your life who has B positive blood and has absolutely no idea what it actually means. Because somewhere out there, right now, in a hospital file, on a blood donation card, in a family medical history folder tucked away in a drawer, that same tiny 4 amino acid mutation is still being carried forward, quietly, exactly the way it has been for the last 10,000 years, waiting for someone to finally understand what it has been trying to say all along.